Welcome to This Explainer. Today we're embarking on what's essentially a biological detective story, and it's taking place deep inside the human body. We're going to explore the incredible architecture of our muscles, unmask the microscopic culprits that literally tear them down, and look at some absolutely groundbreaking research that might just teach us how to flip the switch and stop muscle wasting in its tracks. So, why do our muscles just waste away when we age or get sick? And more importantly, can we actually stop it? Now, muscle loss isn't just an inconvenience for your gym routine, right? It is a critical vulnerability during aging, cancer, AIDS, or starvation. When the body undergoes severe stress, muscle is almost always the first casualty. But why exactly does that happen? Let's find out. Let's put this into perspective with some numbers. In humans, skeletal muscle makes up about 40% of our total body weight and holds a staggering 50 to 75% of all our proteins. So muscle isn't just for movement, it's an essential survival engine. It acts as the body's primary protein reservoir, which gets drawn upon during severe physiological stress just to keep your vital organs running. Okay, let's dive right into this architecture. Muscle tissue is beautifully organized. It goes from the visible muscle you can see all the way down to individual parallel fibers. And actually, get this, here's a truly surprising fact. These individual muscle cells are massive. We're talking up to 100 micrometers thick and sometimes an astonishing 30 centimeters long. That's almost a foot long for a single cell, creating the powerful bundles that allow us to move. Zooming way down to the microscopic level, we find the sarcomere. This is the actual contractile machinery of the muscle. Made of these perfectly overlapping actin and myosin filaments, this structure is what gives skeletal muscle its striking striped appearance. When these filaments slide past one another, boom, they generate mechanical force. It's a highly ordered structure, but it requires constant maintenance. And that maintenance is essentially a constant biological tug-of-war. The size of your muscle fibers depends entirely on the coordinated balance between building things up and breaking them down. On one side, we have hypertrophy, where protein synthesis exceeds degradation, usually driven by mechanical overload or hormones to build muscle. And on the exact opposite side is atrophy, where protein degradation completely outpaces synthesis. So, what exactly happens when that balance tips unfavorably? Well, we enter a biological crime scene known as muscle atrophy. Now, it's crucial to understand this isn't just a passive process of muscles fading away. Not at all. Atrophy happens as a consequence of extreme stressors. Think neural inactivity, systemic inflammation, or fasting. Under these conditions, the body actively initiates specific signaling pathways to dismantle your own muscle tissue. The primary cellular cleanup crew responsible for this breakdown is called the ubiquitin proteasome system, or the UPS. You can think of it as a highly selective biological shredder. Basically, an enzyme cascade, crucially driven by what's called an E3 ligase, attaches a tiny ubiquitin protein to a specific muscle target. The E3 ligase is essentially the mastermind here. It chooses exactly which protein gets marked for death. And once tagged, that doomed protein is shredded into tiny amino acids. Thinking about this shredder in action is pretty wild. Using cellular energy, the enzymes attach that ubiquitin tag, building up a targeted gene. Then, that doomed protein is fed directly into a barrel-shaped structure called the 26S proteasome. The proteasome literally chews the target protein up chemically, spitting out raw amino acids that the body can then reuse or recycle elsewhere. But the UPS doesn't work alone. Enter autophagy, which literally translates to self-eating. Activating really aggressively during nutrient-poor conditions, like starvation, this system physically engulfs entire chunks of cytoplasm and whole organelles inside a double membrane structure. It then fuses with a lysosome to dissolve and recycle the contents. And notably, skeletal muscle has one of the absolute highest rates of autophagy in the body during fasting. Now, you might be wondering, how does the body know to turn on these destructive cleanup crews? Well, it all comes down to a specific set of genetic blueprints called atrogenes. Basically, these are atrophy-related genes that are coordinately activated across all sorts of different muscle-wasting conditions. They encode the very enzymes, like those E3 ligases we just talked about, that the UPS and autophagy systems use to actively dismantle your muscle. So who is giving the actual orders to these atrogens? The ultimate mastermind is a family of transcription factors known as FOXO. When growth factors are absent, FOXO enters the nucleus and directly turns on those essential atrophy-related genes, specifically massive muscle destroyers like atrogen-1 and MRF1. FOXO acts as the ultimate commander, coordinating both the UPS and the autophagy pathways at the exact same time. Which leads scientists to a thrilling hypothesis. What if we just cut the mastermind's communication? Seriously, if FOXO is giving the orders to destroy muscle, what happens if we remove FOXO entirely? To test this out, researchers genetically deleted the 3 major FOXO genes specifically in the skeletal muscle of mice. Then they subjected these mice to fasting, which is a condition pretty much guaranteed to induce severe muscle wasting. Now, the magnitude of these results was staggering. Normal mice experienced severe muscle loss and high rates of autophagy, exactly as you'd expect. But the knockout mice, the ones missing those FOXO genes, they were completely protected from muscle loss and weakness. No way, right? Autophagy was blocked entirely. Just by silencing that one mastermind family, physical muscle wasting was fundamentally halted. But in science, there's almost always a plot twist. While FOXO clearly controls a huge portion of muscle breakdown, analyzing the gene expression revealed something weird. Many atrophy-related genes simply weren't induced without FOXO. But the known ubiquitin ligases aren't enough to degrade all the massive proteins in a muscle cell. The data strongly suggested there were still undiscovered accomplices out there, hidden E3 ligases taking orders from FOXO. Enter the thrilling new discovery, the SMART gene. SMART stands for specific of muscle atrophy and regulated by transcription. This is a brand new, previously completely unknown E3 ubiquitin ligase. Through careful experiments, researchers confirmed that SMART forms a complex, taking direct orders from FOXO to specifically target muscle proteins for destruction during periods of atrophy. It was the missing piece of the puzzle. To validate just how critical this new SMART gene was, scientists conducted a final, super elegant experiment using denervation, which is where a muscle loses its nerve supply. Usually, this causes rapid, severe atrophy. But they knocked down the expression of the SMART gene in vivo. And the result? Inhibiting the single SMART gene significantly protected the denervated muscles from wasting away. It proved definitively that SMART is an absolutely critical gear in the whole atrophy program. And this brings us to an incredibly powerful realization. We've zoomed all the way down from a 30-centimeter muscle fiber to a single microscopic gene. If we can map the exact FOXO signature and selectively block accomplice genes like SMART, we are looking at the foundation for entirely new pharmacological treatments. I mean, think about it. Could we one day formulate a therapy to permanently stop the catastrophic muscle wasting associated with cancer, heart failure, and human aging? The discovery of SMART suggests the switch is definitely there. We just have to learn how to flip it. Thanks for joining me on this explainer, and keep questioning the science that makes us who we are.